Hello, this is part 2 to chapter 2, staining. So we have the first topic under staining is that we would want to create the slide to stain. So we're going to use smear preparation and heat fixing for all of our labs when we're looking at different kinds of microbes. So a smear prep is basically getting some bacteria on a slide, right? So you've done something like this where you use one slide to go over, this is to spread the microbes from your teeth, right? When we did the negative stain. You can also put microbes on with a loop. When you have the bacteria on your slide, you're going to want to heat fix it. So heat fixing is just applying a little bit of heat. Let me show you the picture, right? We're going to move the slide through the flame quickly, not hold it in there like this, but quickly so that we can basically melt the organics of the bacterial wall and membrane to the slide so that it won't run off, like you won't lose the bacterial cells as you rinse the different stains through when you're doing your staining techniques. So when you heat fix, you adhere the slides onto the stage by passing through a flame. You want to be careful with fixing bacteria because if you overfix or hold it on the flame too long, the cells will really dehydrate and they shrink and they distort. So the shape might not be really easy to see anymore. Is it a coccus? Is it a rod? Some distortion always happens when you heat fix. You just can't get away with it. It's going to be distorted slightly, but you don't want to over distort. If you underfix and you pass it through too quickly, then some of the cells may not have adhered to the slide and they can wash off during your staining. So these are just some practice issues that you'll have to go through in lab. And so when you find your bacteria on a slide, you can see that these are 3 different circles that the person put bacteria, so it looks cloudy. These 2 over here are very thick. Now, if you can see the cloudiness of bacteria, you're going to get something like this, which is way too thick. The job that you want anytime you look at bacteria under the slide is basically what shape are they, and then what stain did they pick up, what color. When you have a really tight collection of cells like this, it's hard to see the cells separately, and it's hard to see like exactly what shape they have. Also, if you're using 2 colors, this one's just using 1, but if you're using 2 colors, then sometimes that other color dye can get stuck, just physically stuck between the clumps of cells so that you're going to see 2 colors, and sometimes you don't want that. The middle one is the better one to strive for, where it's a little bit thinner and you want those cells far apart. You want to be able to see individual shapes, and you also want to see the arrangement, right? So this is like the arrangement being, is it a strepto or staphylo, right? Is it cells that make a chain? Is it strepto? Or is it cells that make a clump? Staphylo, right? So if they're really jammed together like this, you're not going to be able to see the arrangements of those cells either. So if it's too thick, the shape of the cell is not being able to be looked at well. The the arrangement of the cell. Sorry for my horrible. The arrangement of the cell is going to be difficult to look at, and also if you're using 2 stains, 2 colors, that other color might get trapped in there, and so it's going to be giving you information that's false. Okay. So we talked about heat fixing. Another kind of fixing bacteria to the slide is it's a chemical fixation, and we can use methanol, which is a kind of alcohol. So you can that will also chemically just fix the bacteria to the slide. Why you would use this is to preserve structures that are easily distorted by heat. So flagella tend to get burned up, pili or any biofilm. There's a structure called a capsule that we will learn about. Some bacteria have a capsule on top of them, or something called a slime layer, which is basically like a sugary, starchy layer on top of them. And if you apply heat, that's going to go away. So if you want to see those two structures, you have to chemically fix, or don't heat fix at all. All right, why are smear preparations fixed? The answer, you can pause it here to answer the question. All right, so it is to kill the microbes and fix them to the slide. Okay, coloring comes from staining. So smear prep is a different thing than staining. So we always want to get the bacteria from the sample, put it on a slide, fix it. That's our smear prep. Then we apply a stain. And the stains are different, and they are, different information can be gained from the stain. So staining. Stains are colors, right? They're dyes, and the dyes have either a positive or negative ion on them, and that color is a chromophore. It attaches to the cells but doesn't bind to the background of the slide. So basic dyes have a positive ion, and acidic dyes have a negative ion. So most bacteria have a slightly negatively charged external wall, and some of them will have an external membrane. So what kind of dye would work best for most bacteria? It would be the positively charged ion, right? Because positive and negative attract each other. This is a nice view of when you have an unstained slide. You can't see the bacteria at all because they're just bags of water, right? Stained, you can see now we have bacteria at 40x. So this is going to be your scanning view. And then at 100x, now we can see this is actually a Diplococcus. It's two small circle cells very close together. So these are the kinds of stains you want to be familiar with. One is called a simple stain. It is used to highlight and stain the entire organism and only uses one color. That's why it's simple, because it's like, bam, one color. What information can we get from just putting a color on the bacteria? We can see the shape of the bacteria. So what shape are these? This is a rod shape, right? Or a bacillus. And how is the arrangement, right? Shape, I should add arrangement. Go ahead and add that to your notes. Forgot that. Okay, so is this arrangement, does it have an arrangement? Number one, yeah, I can see that these cells are making a little line. They're attached to each other, back to back to back. And so that is going to be strepto. Strepto. So this could be called strepto. And then what's the shape? Bacillus. Streptobacillus. So we can, ooh, sorry about that. Streptobacillus. Yeah, and then you can see the relative size. All bacteria are not the same size. So you have really big circles, small circles, long, long rods, short baby rods, so we can see the relative size. And that's it. We just see the shape, the size, the arrangement. Other kind of stains tell us more information, okay? Differential stains means that we're going to differentiate between two things. We can differentiate the kind of cell they have, gram stain, gram positive versus gram negative. We can see whether the stain, whether bacteria have spores. We can have a capsule stain to look for capsules. Acid fast stain is specialized to look for acid fast bacteria. These are bacteria, well, I'll explain it when we get there. And then we can have a special stain just to see the flagella. So differential stains always have two dyes. There is a counter stain because one color will indicate one structure and the other color will indicate something else, the other structure. So we either want to see two cell types or we want to see the parts of those cells. So let's walk through. This is an example of a gram stain. We can see, right, that we have pink rod-shaped bacteria. And then we have a purple rounder bacteria. And so this is going to be pink rods and purple circles, which tell you the information if they're gram-positive or gram-negative, and we'll look at the procedure here. So this is the one procedure that you're going to want to know the order of the stains that go on. So what we do is you apply a purple stain. The purple is crystal violet. And then what we do is we apply a mordant. The mordant is iodine in this case. What does a mordant do? A mordant helps it basically binds. So if this was a piece of my purple, let me give purple. Okay, so this is a purple chromophore. And iodine comes in, and iodine binds to the purple chromophore. Iodine is a yellowish color. Oops. It won't let me do it. So iodine will be blue. Iodine is blue, and it basically just sticks to the purple. So you can see how the whole combination is going to be bigger, right? It's like, it makes it a bigger, bulkier thing. Because the next step is an alcohol wash that decolorizes the cells. So notice here that when we have the purple on, the rods and the circles, the coccus, are purple. We add the mordant. The mordant doesn't add another color, it just binds to the purple to keep the purple inside the cells. The purple stays inside gram-positive cells. And this is due to the types of cell walls, and we will get there in lab. When we rinse with alcohol, you can see that the rods have lost that purple color and the circles have maintained them. So the cell wall of gram-positive cells are going to be thick enough that the whole mordant and crystal violet stay inside them, even when you wash it with alcohol. The gram-negative cells have a different cell wall, and they are easily, it's a thinner cell wall, and the alcohol easily removes the purple. So now we have this difference between our gram-positive cells will be purple permanently, and our gram-negative cells are now colorless. So in order to see those gram-negative cells, we're going to have to put a counter stain on, which is going to be our safranin. So safranin is pink, very light pink, and it goes into the cells that are colorless. Now, it also goes into the gram-positive cells, but here's the thing. Safranin is a lighter color. Purple is really dark. So even though some of these guys are picking up pink, they're still going to look purple because the purple's trapped and it's darker. But the colorless ones are going to pick up that light pink and show up light pink. So when you look at the results of a gram stain and you have a mixed culture of bacteria, we can see we have our pink gram-negative rods and we have our purple gram-positive coccus. So that's a perfect picture of a gram stain. So differential, because it can differentiate the type of cell wall they have. Okay, is it a gram-positive cell wall? Is it a gram-negative cell wall? Okay. So what color are gram-negative cells after the completion of the gram staining process? Pink. What is the mordant? Iodine. All right, acid fast. What is acid fast for? So this is another mixed bag of bacteria. We again see a rod-shaped bacteria and circle bacteria. However, this is not a gram stain, okay? So we're not looking for gram-positive, gram-negative. This is not what it's telling you. If you use acid fast procedure, what it does is it stains acid fast bacteria, have a special cell wall. They have kind of a wax. It is a waxy coating on top of them, and it's called mycolic acid. Let's write it down here. Okay, but wax is really hard to get into, right? And this kind of staining technique shows you what kind of cells have this mycolic acid on their cell walls and which one do not. So you will learn that the pink ones are acid fast, and then these purple ones are not acid fast. So you see how we do have two colors, and we can differentiate these cells based on whether they have that mycolic acid wall or not. A really important clinical bacteria that this is used for is Mycobacterium tuberculosis. Tuberculosis, as you know, is a disease of the lungs, or maybe not tuberculosis, but if you want to do an acid fast stain on someone's sputum, you know, they cough up if they're sick, and then you acid fast stain it and you see pink, that's bad news. That is a Mycobacterium tuberculosis. All right, Mycobacterium leprae should be red. They call it red. It was kind of pinkish, but it's red. Next stain we have is an endospore stain. So endospores are oftentimes shorthanded as spores. They're different than plant spores, fern spores, fungal spores, completely different. Endospores are a dormant structure, okay? And we'll talk about this in another chapter. They're very hardy. They're like the hardest nut to crack. They're basically, when bacteria go dormant, they make a wall around their DNA, and that spore is basically their DNA within this really, really tough protein shell. So their DNA is protected, okay, with a spore coat. So just a really, really, really thick spore coat. Some cells, some bacteria can make spores and others don't. Okay, so if we want to see, hey, does this bacteria have spores? Then we use an endospore stain. And again, with two colors, malachite green is added with heat. Okay, now we have to do something different because those spores are so tough that the color of green does not get in, okay, without heat. So when we add heat, right, heat's going to drive that green color into the spore and color it green. And then safranin will color everything else. So what it'll look like is this, okay? So these are rod bacteria, right, bacillus-shaped bacteria, and we can see the pink where there's no spore, but then everywhere where you have that greenish, it looks like light blue, but it's a green color that you're adding. And you see those green colors inside the middle of your bacillus, that's the spore it's making. So those are going dormant. This is a spore just out in the open. So that is basically a free spore. So spores develop inside the rod, and then when the spore is released, it's a free spore. This is another method to make, it's basically a spore stain also, where you can see that the really, really bright pink areas are spores, and then the bacterial cells themselves, these bacilli, are clear. All right, negative staining is really important as well. So this kind of negative stain is specialized to look at capsules. Capsules, again, are a really thick, you can think of it as mucus almost, like it's a really thick gelatinous layer made of carbohydrates and proteins outside the cell. So it's like a protective extra layer that some bacteria have. If you want to see whether or not a bacteria has a capsule, remember that capsules were one of those things that were really delicate and they do not do well with heat fixing. So you don't want to heat fix this, but what you see is like this stain is going to stain the bacteria, it's going to stain the background, but the capsule does not pick up the stain. So that white halo around everything is the capsule. There's another kind of stain, negative stain that we call, it's a negative stain, but it's with nigrosin, which is what we did in class. This gives you a black or purplish background, and then the cells remain uncolored. They are clear. So this kind of stain, like nigrosin, is going to be repelled from bacteria, which means what charge does it have, right? Nigrosin, if the bacterial cell walls are negative, what should nigrosin's chromophore be? also negative because negative charges repel each other so that the cells don't actually pick up the stain, they remain clear. Okay, so here's a question. Basic dyes are? So basic dyes are positive and they are attracted to most kinds of cell walls because most bacterial cell walls are negatively charged. What is staining? It's going to be answer D, coloring the microorganisms with a dye that emphasizes certain structures. This one is smear prep, right? Killing the microorganism, fixing to the slide, this is the heat fixing step. Distortions of cell size and shape are minimized in. So this is going to be your negative stain. Negative staining usually does not include a heat fix step, and it's very good for not distorting, minimizing distortion. Capsules are stained with a negative stain. We just looked at that. Endospores are stained with this one, Schaefer-Fulton stain. All right, so this is another image of heat fixing. I think we don't need to talk about this. And positive, negative, this is just a review, right? So this is a positive stain where we have the cells colored, but this is a negative stain where we see the cells not colored, but the background is. This is the gram stain procedure. So you want to know the steps of this, and we'll get into the cell wall in detail in the next chapter. And this is another image of the gram stain. It's not as perfect as the other ones we looked at, but if you study carefully, you can see gram-negative rods, and you can see the purple coccus-shaped bacteria. Here's another image of the acid-fast stain. This is a little bit more lifelike because the rods really kind of stick together. But anything that's red here that's picked up with acid-fast, that's going to be a Mycobacterium tuberculosis. Here again is a negative stain, right? We have a nice capsule here. And then here's another spore stain. Flagellar stains, we won't be doing this in class, but there's special stains to pick up flagella. So we'll look at those prepared slides in class. And that's it. So I think I've covered everything on your review list, and good luck studying this chapter.